Yeah, so today we will be discussing about calorie restriction. So calorie restriction essentially is uh, taking less calories, limiting your calories, but not depriving yourself of the nutrients, right? So what essentially happens, some molecular processes, they are changed essentially, which in this context, we'll talk about aging. And in general, this will pave our way towards epigenetics. We'll talk about epigenetics here. So anybody who knows about epigenetics, what is epigenetics?
Epigenetics is changing the expression of a gene without mutating it by causing certain changes in its structure and conformation and in association with the histone proteins.
That is correct. That is correct. So essentially, if there is no change in the sequence, no change in the genome per se, but there is a change in the expression of the gene due to various other factors. It could be modification, it could be like methylation, acetylation, histone modifications essentially. It could be other kind of things. It could be production of some non-coding RNAs. That could also, if I mean, see, this could happen like at the stage of transcription. It could happen post transcription. So these kind of regulations are always there in your body, so that different kind of cells, they behave differently. The reason behind it is epigenetics. So let's dive into this paper. We'll be discussing this: calorie restriction reduces oxidative stress by sirtuin mediated SOD2 activation. So essentially, what happens here is whenever this ad lib is basically ad libitum, that means this study has been done in mice. So when we call ad libitum, that means the mice are free to eat whenever they want to eat. I mean, there is no kind of restriction as such. But when we're talking about calorie restriction, there are limited, meals, and there is a time gap between those meals. So what few things have been observed: the levels of HNE, protein carbonyl content, and GSH and GSSG, which is essentially glutathione levels, which are markers for the oxidative state of the organism, of the cell in particular. So if we see the HNE levels, when we are, okay, again, we see sirtuin 3. This is one protein. We'll talk about this protein in detail today. So this protein is either present in the mice or they are knocked out. So you guys, I hope you know about what is knockout of a protein, right?
Yes, sir.
Okay, good
So
enough.
can you tell at once
Yeah,
what's
so
knockout?
knockout is nothing, just for the understanding of here. Knockout means that that protein will not be expressed, right? So you knock out, you remove that gene, or you interfere, you can add some elements, some other element into the gene, so that that protein becomes non-functional. So there are various ways of doing
Okay,
that.
okay.
We are not discussing how it is done, but essentially,
Mutagenesis.
yes, mutagenesis could be one way. There could be other ways as well. You must have heard about Krelog system. I'm not sure if you have read about it, but anyway, many other systems. There could be conditional knockouts. There could be anyway knockouts. Like if we have talked about previously also, when certain genes were inserted or certain sequences were inserted, the open reading frame, it changes and the protein is no longer produced. So it's as simple as that. So let's not talk how knockouts are made, but knockout is made in this case. So sirtuins are knocked out. So we will see the role of sirtuins very soon. What happens is, when the mice is given a diet where it can eat anything it wants to eat, so there is a increased level of HNE, which is a marker of lipid peroxidation. While when there is calorie restriction, we are only comparing the wild types here. So when there is calorie restriction, the HNE levels go down. So HNE level is a marker of lipid peroxidation. Essentially, it gives you how much ROS is produced, peroxidase or peroxides. These are ROS, reactive oxygen species. We have already talked about it. So if the HNE is lowered, that means the ROS levels, they are also lowered. We are only comparing the wild type for now. But when we are doing the same experiment for knockout cells, SIRTwin3 knockouts, SIRT3 knockout cells, then the change is not that evident. I mean, bar, this bar, and this bar doesn't have a significant difference. Is that clear to everybody?
Sir, it last, but please explain once more. How to
SIRTwin3, when it is not present in the cell, when this is knocked out, right? In the absence of SIRTwin3, whether the mice goes for calorie restriction or it has a normal diet, there is no effect on the ROS levels.
Okay, sir.
So what can you infer from this?
SIRT3, it actually controls the
That is correct.
reactive
That is
oxygen
correct.
species.
Yes, that it does. So we will see. So essentially the other graphs, they also show the same thing. So I'm not going to explain everything, the same thing 3 times. But essentially what you said is right, that SIRTwin3 has some role in managing, or I would say tuning the ROS levels in the cell. So we'll see how that happens. So first we'll talk about what is a SIRTwin, what are SIRTwins. So sirtuins are deacetylases. So you know what deacetylases are? What do they do?
Removing the acetyl groups.
That is correct. That they remove the acetyl group. So as you can see in this cartoon here, so essentially SOD2, it remains in an acetylated state. It remains in an acetylated state in normal condition. While it is in its acetylated state, it is non-functional. SOD2 is non-functional when it is in its acetylated state. Is that clear till here?
Yes, sir.
Fine. Now these sirtuins, they deacetylate these enzymes SOD2 and then it becomes functional. So whenever there is calorie restriction, there is an increased production of sirtuins, essentially sirtuin3, that causes activation of SOD2. So if there is higher activity of SOD2, there will be less reactive oxygen species. Is that clear to all of
Yes,
you?
yes, sir.
Okay, so far so good. Then essentially we will see what happens. So the same thing is written that SOD2 is present in acetylated form, it is active in its deacetylated form, and sirtuins are NAD plus dependent protein deacetylases. So whenever there is calorie restriction, what we see that sirtuin, it binds to SOD2, it deacetylates it, then the activity is increased, SOD2 activity is increased, and there is reduction in reactive oxygen species. Is that clear to all of you so far?
Yes, sir.
Okay, then let's see. So here what we are seeing, here we are going for SOD2, I mean sirtuin3 knockout and wild type. Wait, let me have a look. Okay, SOD2 is either present or it's not present. So when SOD2 is not present in a cell, when SOD2 is knocked out, in this case, either SOD2 is present or SOD2 is not present. When SOD2 is present, when sirtuins are not present, there is a higher amount of ROS levels. But when sirtuins are present, there is a lower level of ROS level. So essentially what we saw previously, that sirtuins are important for deacetylation of SOD2, and then in its increase in activity, that is what is confirmed from this graph. When SOD2 is knocked out, SOD2 is not present in the cell, whether sirtuins are present or they are not present, it does not matter. The reactive oxygen species levels, they remain almost similar. So sirtuins by themselves cannot reduce the reactive oxygen species levels, but they actually deacetylate the SOD2 enzyme and they reduce the levels of reactive oxygen species. So what they did, they mutated the catalytic site of sirtuins. This is a mutation, this is an amino acid change. So when they mutated the catalytic site of sirtuins, what they observed that when sirtuins were non-functional, then the SOD2 activity was reduced. When the sirtuins are functional, it shows the highest SOD2 activity in the middle graph. Is it clear? Or is there any confusion?
Sir, what is this SIRT H248Y? This one.
That is a sirtuin mutant. So this is a mutant where this is an essentially H248, that is histidine 248. It's an essential catalytic site of this enzyme. I mean, it is a deacetylase, it's an enzyme. So this is an important catalytic site of the enzyme. So if we mutate this to Y, this is Y is what, which amino acid?
Tyrosine.
Yeah, so when we mutate histidine to tyrosine, then the catalytic activity is lost. So the enzyme is non-functional.
Okay, then simultaneously the ROS concentration increases.
This is SOD2 activity levels, it decreases.
SOD2 activity level decreases.
So now let's talk about acetylation in general. Usually lysine residues are acetylated. That I hope you guys know. Lysines K, like here you can see K53, K68, K is all lysine. So usually lysine residues, they are acetylated. So in the B panel, what you see here is the multiple sequence alignment of this protein. So here we are seeing SOD2 protein. So the K, I mean, this is 53, location 53, and this is 89. They are relatively conserved in all these species. These are different species. These are different organisms. This is like Homo sapiens. Then this is the next one is Mus musculus. So similarly, these are different species. Mus musculus is essentially mouse. So in different species, we see that K53, K89, these kind of residues are conserved. These residues are also close to the active site, like this histidine. I mean, if you see here, these histidines are active sites, so they are also close to the active site. So what what is an active site?
The site on the enzyme
Yes, sir.
where the actual reaction takes place.
So
Good enough.
where substrate binds and fits like a key to a lock, and finally
Yeah,
the
that's good
reaction.
enough. That is good enough. So what they did, they changed these residues from lysine to arginine. So if they change it from lysine to arginine, they could not be acetylated any further. Fine. You understand the logic behind this?
Yes, sir.
So when they mutated this, so if you see the this is a Western blot, you know what is a Western blot. So when they when they made an antibody against acetylated SOD2, they observed when this mutation was there, K5389R means both 53, K53 and K68 both were mutated to arginine. When they did that, so the levels of acetylated SOD2 decreased compared to wild type. Is that clear?
Yes, sir.
We see the same thing here also. So when there is wild type SOD2, then in a control setup where there is no sirtuins, there we observe that the relative SOD2 activity is the lowest. When sirtuins are given in this, sirtuins are overexpressed, at that time the SOD2 activities is increased. When we use SOD2 mutant, which has K5389 mutated to arginine, lysine mutated to arginine, we see an increase in SOD2 activity. Can you tell me why?
Sir, repeat the question once more.
When we are doing this mutant K5389R, it shows the highest SOD2 activity. Can you see this bar graph?
Yes, sir. What is R? K is lysine, R is arginine, right? Okay.
So lysine to arginine is the change which we are doing here.
Then SO2 activity increases.
Hmm.
Sir.
Why so?
Because if lysine is changed into arginine, uh, the amount of acetylation will be more. Like,
More or less?
sorry, the acetylation will be less. Sorry, because it is converted into arginine, so the
So
acetylation will be less.
arginines, they are not acetylated, whereas lysines are. So when we are changing lysine to arginine, acetylation is not possible. So hence the protein in its native form remains in a deacetylated state. So no matter whether you give sirtuins or not, it will show a high activity. Is that clear to everybody?
Yes, sir.
Okay. So good. Then we also see that when the ROS levels were seen for SOD2 knockouts and the normal SOD2 in the presence of sirtuin, I mean overexpression of sirtuins and in normal condition. So when sirtuins were not overexpressed, there was a high level of ROS. Fine, fine so far.
Yes, sir.
Okay. When sirtuins were overexpressed, the ROS levels decreased. Is that clear?
Yes, sir.
You know why it happened? Because of the deacetylation process.
Yes, sir.
Now, when SOD2 was overexpressed, okay, when SOD2 was overexpressed, then the reactive oxygen species levels, they decreased.
But sir, if the SOD2 activity increases and but the amount of sirtuins they remain constant, it would not. It shouldn't have affected too much, right?
Yeah, that's what you are seeing. Is it a very different change?
Okay.
So what you are thinking is right. So that's why the change is less compared to what you see here. When sirtuins were overexpressed, reactive oxygen species, they dropped significantly. Is that clear?
Yes, sir.
Okay. So now when we did this mutated version of SOD2, then there was a very low number of I mean reactive oxygen species, whereas in presence of sirtuins, the number went even lower. Is that clear?
Yes, sir.
Yeah, fine then. Anything else that you need to know here? Let me look here. I don't think. I mean, even if you see the number of live cells in the right G graph, here you see it is just the opposite of the ROS levels. If you see E and G, they are very similar. It's just opposite. So what happens? The number of live cells, they increase when you have more amount of SOD2 or you have more amount of sirtuins. Clear on that?
Yes, sir.
Fine. So here also what they did, now they are doing calorie restriction. Fine. Calorie restriction. So you know calorie restriction, what it is? Limited calories are given. Uh, there is a time interval between each meal. So when calorie restriction was done in wild type, where there was active sirtuins, there the SOD2 activity was the highest. When sirtuins was knocked out, SOD2 activity, it reduced. Could you say why that happens?
Because means during calorie restriction, the amount of sirtuins they increase, which deacetylate the SOD2 enzymes, for which reactive oxygen species gets down. But when the CR, I mean, the ये की बोले, sirtuin gets knocked out, means it gets mutated or changed or becomes non-functional, then less amount of deacetylation occurs. So therefore, less amount of SOD2 active SOD2 enzymes will be present, which will cause a lower amount of SOD2 activity.
You are right. You are right. So let's go back to this slide. So we can say, I mean, this is we can say this is the summary essentially. So when calorie restriction is there, there is an increased amount of SIRT2 SIRT3, and SOD2 is deacetylated and its activity increases. When there is SIRT2 knockout, deacetylation is not possible. Hence, the SOD2 activity, it remains low, and there is higher amount of reactive oxygen species or oxidative damage. Clear on that? Is everybody clear?
Yes, sir.
Any questions so far? Anybody? Okay, cool. So let's go back. Let's talk about epigenetics. So at this time, if you see the timeline, so aging and associated epigenetic regulations, it started, we started discovering them from 1967, where we understood that epigenetic regulation is also involved in aging. You must have known about epigenetic regulation in cancer as well. So yeah, both things happen. And even therapies with respect to changing the epigenetics and delaying aging were also discovered. So this is the timeline. So in 1935, this paper, I mean, the calorie restriction was discovered. After that, there was certain activating compounds. So that makes sense because sirtuins will essentially deacetylate SOD2, and it will increase its activity. It will reduce the oxidative damage in a cell. So circadian rhythm reprogramming, senolytics. You know what is senolytics?
No, sir.
Senolytics, it comes from two terms. One is senescence, another one is lytics, essentially. So
Oh,
senescent cells, they are killed. So these are some antibodies, essentially dasatinib and quercetin. They target the senescent cells, they kill them essentially. So this is senolytic therapy is also very active in age delaying process or age reversal, as we call it. And there are some aging vaccine therapies also. It's very new actually. So we'll not be discussing about that. So when we talk about the methylation status, so what happens when there is a methyl group in the promoter region if the DNA is methylated?
The polymerase cannot bind to the promoter.
So what will happen?
Transcription turned off.
So there will be a repression, right? So this part is clear. If the DNA is methylated, there will be less amount of expression of the protein, right?
Yes, sir.
Fair enough. So with age, what we see, if you see this figure, this cartoon over here, with age, what happens is that the methylation status, it changes. Usually it decreases. So when the methylation decreases, genes are expressed. So how would that be a disadvantage?
So repeat the question.
Methylation decreases with aging. Some genes which were not previously expressed are being expressed. So how would that be detrimental?
Because there might be some genes like the apoptotic genes which actually cause the cell to undergo lysis.
Yes, yes. There is chance that there might be activation of apoptotic genes. You're right. There is chance of autoimmune diseases to come up
Diseases.
during aging, right? So all these things are finely regulated by the methylation status or the epigenetics. But if it is changed, then there might be some problem. In a similar way, how could cancer be developed if there is a change in epigenetics? So what would happen? I mean, which kind of gene would undergo hypomethylation?
a gene for telomerase enzyme or shelterin.
What else? I mean, which particular gene do you think is
P53,
responsible?
P53, P53, P53.
P53. Okay, P53 is a tumor suppressor gene.
Yeah, so if it gets like I don't know if
If the
it gets
methylation of
like,
P53 is reduced, what would happen? P53 will be expressed more. So that's a good thing.
then in that case there will be more methylation of that P53. If occurs, then it might cause cancer.
Yeah, so essentially, let's see. So this is another paper we have taken our two graphs, very simple. So they just see the methyl cytosine levels. Fine. So these are normal cell lines. These are immortal cell lines. So immortal cell lines, they never die essentially. So when a cell line could die, we see that the methylation status, it decreases with population doublings. You know what is population doubling?
Yes,
Like
sir.
double, yes.
The amount of time a culture need to double.
Right, that is correct. And passage number is essentially the number of times you have subcultured your eukaryotic cell. So what happens in immortal cell lines? We observe that their methylation level, they do not change. So this very particular, faithful, replication of the methylation status, that happens due to the epigenetic clock. So when you age, this epigenetic clock is disturbed. So what happens? Let's say you have neuronal cells, you have liver cells, right? So liver cells, they have more proliferative, or let's say you have, okay, let's say, so neuronal cells, they will have less proliferative potential, and your hepatic cells, they will have more proliferative potential. So when they are proliferating, what could happen? That the epigenetic signatures which are present, which make them hepatic cell, that might change. Are you understanding this? Or is it
Yes, sir.
just going over your head?
Understood, sir.
So if the epigenetic signature they change, there could be something which we call de-differentiation. There could be formation of something which we called as cancer stem cells. So anybody knows anything about stem cells?
Yes, cells which have the ability to divide and differentiate into any type of cell.
Right, you are right. And essentially they have unlimited replicative potential, any kind
Yes.
of stem cell. So they are important for regeneration. So whenever there is a lack of any kind of cell type, if there is some injury, these cells will essentially differentiate. If there is a normal stem cell, let's say hematopoietic stem cell, HSC, they will differentiate, they will into different types of blood cells. So it ranges from RBCs, different types of WBCs. Fine. So these kind of things might happen. So this differentiation process, it is also closely regulated by epigenetics. Is that, is that understood by everybody?
Yes, sir.
So the epigenetics will ensure that this cell behaves in the way it is supposed to behave. So if there are genes that are associated with, hematopoiesis, those kind of things, how the cell morphology will be, different types of cells, they have different kind of morphologies. All these things are controlled by epigenetics. So during each replication process, from hematopoietic stem cell to RBCs, we want that the epigenetic signature to remain intact. But if it changes, let's say if it changes in between, then what will happen? It will lead to formation of, let's say, cancer stem cell. So there will be formation of RBCs, WBCs, but their function will be very different. They will be non-functional as such. So is that clear?
Yes, sir.
We will discuss about it again. So let's come here. So we have talked about lamins, right?
Yes, sir.
So lamins in a young cell, they remain intact. Fine. In a young cell, they remain intact, and all the the closely condensed elements, the closely condensed gene, they are closer to the lamins, so they are never expressed. But when a cell becomes old, the lamins they they lose their integrity. So these genes which are usually not expressed, they are now expressed because lamins are not present. So what happens? Uh, there could be a lot of things which could happen. So there could be, like, let's say, oncogenes. So oncogenes, they have not only they have some epigenetic regulations also. So oncogenes usually they will remain as proto-oncogenes. You know that oncogenes are usually they are not expressed. So some of their one of the way is epigenetics also, where it is heavily methylated. The histones are heavily methylated and they are closer to the lamins so that they are not expressed. But now, given that the cell is old, the lamins are degrading. Now these oncogenes they could be expressed. So what would happen then?
Cancer.
So that is how aging causes a lot of diseases to come up. It's not only cancer, a lot of things, even cancer is related to aging. Many types of cardiovascular diseases, they are related to aging. So I hope I could like make you explain how epigenetics regulates all of this. So while we are talking about epigenetics, so there could be two things which could happen. I mean, epigenetics, the methylation status, it might increase with age. The methylation status, it might decrease with age. The methylation status might not have any relation with age. So when there is no relation between inconsistent relationship between age and the methylation status, we call it as an epigenetic drift. When there is a consistent relationship, this could be like this could be like this also.
Eh? So,
Yeah. So when there is a consistent relationship, then we call it as an epigenetic clock. Is that clear?
Yes, sir.
So epigenetically, I mean, let's say all of you are of the same age group. You are having the same calendar age, almost same. I mean, all of you are like 19, 20 years old. Is that correct?
Yes, sir.
But if you see the epigenetics, individual epigenetics, then maybe some of you have biologically aged more than the rest of your classmates. Some of you might look younger. So see, epigenetics is involved behind this process. So if there is any change in epigenetics, that could lead to, I mean, epigenetics means methylation, acetylation, other kind of regulations apart from genetics. So genetics could change, epigenetics could also change. That's the point. It's a very novel field. So here they are actually, here they discuss that Yeah. Am I audible to all of you?
Yes, sir.
Okay, where did you lose the
Yes,
connection? I mean,
sir.
where did I lose the connection? I'm really sorry for that. Which slide were you did you see last?
Epigenetics in aging.
15.
This one?
Previous one.
Previous one. So yeah, as I was telling you, I'm really sorry for that. Sometimes there is some network problems here in IITs. It happens. But anyway, what I wanted to tell you that all the hallmarks that we have discussed so far, sometimes these are also regulated by the epigenetics of the organism. Let's say if the genes they remain in the heterochromatin state, then they are closely compacted together. They are resistant to DNA damage. But if they go to the euchromatin state, so you know what is a heterochromatin and what's a euchromatin? These are chromosome remodeling
Yes,
concepts.
yes, yes,
Yes.
sir.
So when they are in the euchromatin state, even they are more prone to damage as well. So when I say that oncogenes, they could be, they might be in a condensed state. So these they remain as proto-oncogenes. They are not mutable. But once they change to a euchromatin state in a relaxed conformation, they are more prone to mutation. Is that clear to all of you?
Yes, sir.
Yeah, fine. Then what we talked about was epigenetic clock, aging clock, essentially, with respect to calorie restriction. So what happens when there is calorie restriction? There is also an increase in DNMTs. You know what is DNMTs? Hello.
Yes, sir.
Yeah. DNA methyl transferases.
When does the level increases, sir? Please repeat once.
When there is calorie restriction, there is an increase in DNMTs as well. So these DNMTs means DNA methyl transferases. They maintain the methylated state of the cell. Clear?
Okay.
So we have already discussed about sirtuins also, histone acetylations. Sirtuins are deacetylases for proteins. Histone is also a protein. So histone deacetylation is also controlled by sirtuins. So we have already seen how calorie restriction has an effect on sirtuins, right? So this part is, I hope it is clear. So
Yes.
further, then there are some therapies like small molecules we can reprogram. So epigenetic drugs could be given. Reprogramming could be done. There could be senolytic therapies or some active health changes like calorie restriction is you must have heard it as intermittent fasting. So even using keto diets, it's very popular. Ketogenic diets nowadays, if you some of you if you go to the gym, people might be knowing about all this. So they produce less amount of oxidative reactive oxygen species, so that way it is better. So having a good exercise routine, a good circadian rhythm, even if you are staying awake for long hours in night, it's going to affect you badly. So all these things are related to aging. So any questions so far?
No, sir.
Okay, then I think that's all for today. We have right now two people from NIT Raulkela. So I would like them to just briefly introduce themselves, Ratul and Shashwato, and please interact with your juniors. Hello, is this still going on? I'm not sure.